Hello, everyone. So thank you for joining this webinar on microencapsulation using 3D printing. And I'm very honored that I have been invited to give this webinar together with my student Brenden Moeun and also Craig Bundt at the University of Otago. And we welcome you to participate. I know that this is a remote webinar. I know that we would all like to be together in person, but the beauty of this is that everyone around the world can join, so this is great. So during the webinar, just a couple of housekeeping items. The first one is that if you have any technical problem, please post your problem in the chat window, and Brigitte Hoesli will be happy to help you with your technical issues. But if you have questions on the actual scientific content, please post those questions as we go along in the Q&A window, because we will start answering questions as we go in the Q&A, and also there will be a live Q&A at the end, and we will try to cover all the questions that were asked. If you're interested in past webinars or upcoming ones, please go on the bioencapsulation.net website. The recordings are posted, and today's webinar is also being recorded and posted later. This webinar is organized jointly by the Bioencapsulation Research Group and Encap for Health. The Bioencapsulation Research Group is a large organization, over 7,000 contacts from 80 countries. The BRG organizes industrial meetings and international annual meetings as well. So for more information, please contact bioencapsulation.net. There is also training schools and things like that that are of high interest. And the organization of these webinars, amongst other events, is co-organized by Encap for Health. And so this is part of the training program of that network in Europe. So there will be two portions of the webinar. The first one is going to be more on health applications of 3D printing as it relates to microencapsulation. And that portion will be given by myself as well as Brenden Moeun. And then the second portion will be given by Craig Bundt. So in the first portion, I'm just to present myself. I'm an associate professor at McGill University. I hold the Canada Research Chair in Cell Therapy, Bioprocess Engineering, and we work on stem cell culture and encapsulation, especially for the treatment of diabetes and cardiovascular disease. Brenden Moeun is a student who has been there for a few years. He's at the PhD level. He is funded by a NSERC PhD scholarship, a very prestigious scholarship in Canada. And his expertise is on rheology, mechanics, and 3D printing technologies, as well as cell culture for diabetes. And if you want to check out his publications, please read those excellent scientific and review papers. So with that, we will launch the first portion of the webinar, and don't forget to ask your questions in the Q&A. Hello, everyone. I'm Corinne Hoesli, and I'm an associate professor in the Department of Chemical Engineering at McGill University, and I'm also the Canada Research Chair in Cell Therapy Bioprocess Engineering.
And hi, everyone. I'm Brenden. I'm a PhD candidate in the Department of Chemical Engineering at McGill University, and I work in the lab of Professor Corinne Hoesli.
Today, we're happy to talk about 3D printing for biomedical applications in the context of microencapsulation. And I know many of you joined because you're excited about 3D printing and all its different applications. Today, we will really try to cover what is more related to encapsulation and applications thereof. By the end of the webinar, you should be able to list some of the main applications of 3D printing in the biomedical space as it relates to microencapsulation. You should be able to categorize 3D printing technologies used in medicine, and with a focus on bioprinting, which is printing live cells, because this is some of what we do in our lab. So we actually focused our talk on the printing of living tissues. You should also understand some of the theory behind 3D printability. And then finally, we will cover 2 case studies of work we're doing in our lab on 3D printing for generating these vascularized tissues. When you think about 3D printing in medicine, you can envision all sorts of different applications. It's a very broad field, and we won't be covering all of it today because our focus is on encapsulation. But in general, you can think about dental applications. When you go to the dentist and maybe you get a scan of where you want a replacement part or tooth, and then you can possibly get 3D printed, or at least the mold could be 3D printed, things like that. You can also think about orthopedics or stents and catheters, where you want to adapt the device geometry to the physiology of the patient. You can also think about contraceptives and caplets even that are 3D printed. But not all of these things relate to microencapsulation, right? So why would you want to combine bioencapsulation and 3D printing? So encapsulation generally is used to protect an active ingredient from the environment or to control its release. So these are generally, this is not exhaustive, but generally what encapsulation is used for. Whereas 3D printing really provides control over geometry and allows multi-material assembly. So these are some of the applications where you kind of have a merge of that Venn diagram between encapsulation and 3D printing, right? So again, this is not an exhaustive list, but one example would be if you want to combine 2 active ingredients, but they are incompatible, so they don't need the same microenvironment to sustain their activity. So you might want to print a tablet or a device with multiple compartments that allow you to combine those ingredients. Another example is tissues, living tissues, where you might want to encapsulate the living tissue, but still have it function in a certain way. So the example shown there is hepatic tissue, where you could have hepatocytes. So this is liver tissue, hepatocytes that are detoxifying blood that is coming in another compartment. So you might be using combining bioencapsulation and 3D printing for that kind of application. You could also use 3D printing to generate objects with high surface area, right? Because you can generate intricate geometries, and that can increase the release kinetics because you have more contact between, let's say, a physiological fluid and that printed tablet. Today, we'll focus our talk on creating living tissues, which is research ongoing in our lab and others. And in that area, there's 3 main approaches for fabrication using 3D printing. One relatively simple approach, which actually decouples the printing part and the encapsulation part, is to first 3D print a mold, typically out of a sacrificial material. So it's only a temporary mold, but the 3D printing capability allows you to make complex geometries. And then you cast your cells into that mold. And then you can remove the sacrificial material and be left with a reverse of the original complex geometry. Another example is layer by layer 3D printing, and probably most of you in your mind when you're envisioning 3D printing, you're thinking about additive manufacturing, which is adding things layer by layer until you get your complex objects. So you can do that as well with living cells. And then another example is more like using a support system or starting with a material where then you create some kind of localized structures via 3D printing. And this could be, for example, by embedded writing, where you could actually like extrude material or deposit material into a bath. Or another example of in situ fabrication would be if you use light to create those features to polymerize locally material within that bath. So those are some examples. In terms of the actual 3D printer, like what kind of equipment you need to do this, for bioprinting, which is printing live cells, there's 3 broad categories of printers that are out there available commercially. The most common is extrusion bioprinting, where you have a pump, a syringe pump, or you could have a solenoid valve or something that pushes material through a syringe. It could also just be air pressure. There's droplet-based techniques, which are very similar to the printers that you have at home for, laser or inkjet printers. So the concept is quite similar, where you have kind of an ink that is being projected onto the surface, except that we talk in that case about a bioink because the ink contains living cells. And another example is laser-based or light-based techniques where, as I mentioned before, you can use light to create a reaction in situ by focusing your light onto a certain spot. These are some examples of bioprinters that we have available at McGill. So we've got in our lab RegenHue 3D Discovery, which is based on extrusion printing. We also, with Luc Mongeau, have together a bioassembly bot, which is a 6-axis robotic arm that can approach the object that you're printing from different angles, which is quite unique because it allows you to make curved geometries and things that are difficult to print via layer by layer kind of assembly. And I don't know if some of you have seen Westworld. That's the kind of 3D printer they're envisioning in that show. There's also a lot of other extrusion printers that are a little bit lower end that are available on campus, but basically these are very convenient because you can place them easily into a biosafety cabinet, and they're great for rapid prototyping. We also have on campus a droplet-based system that I mentioned earlier, the GSIM that's shown there. And we have an RX1 printer, which is based on microfluidics. So it's also a type of extrusion, except that it's based on flow focusing. So this is not all the printers that are available, but those are some of the technologies we have on campus. So why would you want to use 3D printing for encapsulation? Maybe it makes fancy objects. It's a hot topic. It helps you get funding because reviewers get excited about using 3D printing. It helps you recruit great grad students interested in 3D printing. These, in my vision, are all bad reasons for using 3D printing. Actual reasons why you should consider 3D printing is because you can fabricate multi-material objects. It gives you rapid prototyping capability. You can make geometries that are difficult to achieve otherwise. And in certain cases, it allows you to achieve more precision and reproducibility, but that's not always the case. So I like to always think about, can I make this with a pipette manually? If so, maybe 3D printing is not the right approach for what I'm trying to do. there's a lot of challenges when you get into the 3D printing or into the bioprinting field, so printing of live cells, but 3D printing in general. So how to determine the geometry that you want to print, and then how to convert that geometry into something that's actually printable. Often you have to slice it and make something that you can deposit layer by layer, for example. You have to consider the cell compatibility with your bioinks or your printing strategy. And that also goes into material selection. So your material you might want to confer certain properties to the material to make it cell compatible and printable. And then finally, it's very difficult often to monitor success. So how do you make sure that the geometry you obtained corresponds to the geometry you wanted, but also how do you keep that tissue alive during 3D printing and after?
Great. So now that you have a good idea of the background around 3D printing, I would like to talk to you about how you can create your own 3D printing material, or how can you implement 3D printing for your own research. So here, let's talk about the general principles of printability, or materials that you can 3D print. So generally, when you are extrusion 3D printing anyway, you have a barrel and a nozzle, and you have a material inside of your barrel that's being extruded through your nozzle. So this is a blue material here. And there's a force generated either through air or through a piston, like Professor Hoesli had mentioned. But there's a force here that's pushing the material through the barrel and through the nozzle. Now, when the material is being pushed through the nozzle, essentially what you want is something that behaves like a liquid. So this is important because you want your material to be able to flow out of your nozzle very easily. If you have cells in there, it'll minimize the stresses that are imparted onto the cells and helps you keep your cells alive. However, once the material is done being extruded or is deposited, you would like it to behave like a solid, and not just slowly become a solid, but immediately freeze or solidify in order to maintain that 3D shape that you're trying to achieve. So in other words, a key factor in 3D printing is the rapid solidification. So going from a liquid to a solid. Now let's talk about how we can achieve this. So there are a lot of different ways you can do this using curing agents, depending on your environment, depending on your configuration, different chemicals, UV sources, for example. However, two ways that you can achieve rapid solidification, or how we have done it in our lab, is through a rapid change in temperature and the engineering of a thixotropic material. So we'll get into what thixotropic means later, but these are the two case studies that we'll be focusing on today. Now, our first talk is about using a rapid change of temperature to create, to be able to 3D print sugar. Now, this is a team that we have been working with very closely for many years in order to develop this technology. So we have Professor Lees from McGill, Dr. Paraskevas from the McGill University Health Center, Dr. Marc-André Fortin from the Université Laval, and André Bégin-Drolet, professor, Dr. Ruel, and both of these people are from the Université Laval as well. And this work has been largely funded by TESEL, JDRF, and Diabetes Quebec, or Diabetes Quebec. Now, our goal was to 3D print sugar, and this is the solution that we came up with. It's a customized 3D printer. The 3D printer you can get commercially for pretty cheap, but it was engineered in order to be able to 3D print. Now, I won't go through all of the different customizations, but here are the main ones that pertain to 3D printability. So first, we talked about having the material behave like a liquid as it flows through the barrel and nozzle. And this is achieved through temperature control. So the temperature of the nozzle and the temperature of the barrel are controlled in order to maintain a liquid behavior of the sugar or of the caramel. Now, in order to get that rapid solidification, we have these cooling air jets here. So those are the structures you see there. And what those do is they shoot cold air at the material as soon as it's deposited. So this convection improves the heat transport of the material, making it be able to transition from a liquid to a solid. And as mentioned, the temperatures chosen in the barrel and in the nozzle are controlled, and they're chosen based off of the viscosity and the glass transition temperature of the sugar glass. So this is the point or the temperature at which the material changes from a flowing behavior to a strictly solid behavior. Now, how do we print something that we want from this ink? So we have this ink, we have this printer that can 3D print things, but what's important about 3D printing is the accuracy and the precision and really achieving the geometry that you want to. So you can, for example, get the triangle or the square that you want, but the size matters, the dimensions matter, the micro details matter. So this all boils down in extrusion 3D printing anyway to the diameter of the printed filament. So if you go back to our 3D printing head, we have this filament that's being 3D printed, and what really matters is the diameter here. Or that's a big part of a lot of 3D prints. That determines the scale of your overall print and also is the limiting factor in the details in which you're able to print. So based off of mass transport or mass conservation, you can come up with this equation here that describes how the diameter of your printed filament is a function of your printing parameters. So it's more specifically, dB, for example, is the diameter of your barrel. So this is something that you can choose, something that's preset perhaps in your 3D printer, but also this diameter is a function of the extrusion speed. So the extrusion speed is the speed at which the material is being deposited through the barrel. So this is related to the flow rate. It's essentially how fast your piston is pushing down on your through your barrel if you're thinking about a syringe. Now, another speed that's really important is the feed rate or the print speed. So this is VF, and that's the speed at which the whole printing structure is translating. So that's the barrel and the nozzle. While it's extruding, all of it is being translated at a speed VF. So the diameter of what you're printing is all related to these parameters. We also have some correction parameters for material compression and expansion, which is C, but also for a more empirical or random error, which you will see as B. So in order to be able to print what we wanted from the sugar, we created a relationship of the printing parameters to the resulting diameters, the resulting print diameter based on this model. So you can see that it follows very closely this mass transport theory. And in order to evaluate how good or how great or how accurate our print was, we used micro CT in order to compare a CAD model, which is what you see in red, with what we ended up printing, a 3D scan of it anyway in blue, overlapping it, we can see where they match and where they don't match. And then going further, we were able to quantitatively determine the differences between the two items, so the CAD model and the 3D print. And we were able to do this with the help of Professor Marc-André Fortin at the Université Laval. And this hopefully gives you an idea of how you can characterize your 3D printing through this sort of diameter control, and also how to evaluate your resulting prints. Now, a lot of talk about 3D printing sugar. So why would we want to do this? Well, this goes back to the technique that Professor Hoesli had talked about, and it's the 3D print and cast method. So this turns out to be a really great way to vascularize artificial tissue or tissue constructs. So as you know, sugar is pretty widely available. It's also largely cytocompatible. And as hopefully we've shown, it's relatively easy to 3D print, and you can 3D print it's a very special ink because you can 3D print overhanging constructs, cantilever filaments, things that you aren't normally able to do without a support. So how this technique works is that we 3D print a sugar glass construct. We cast the cells. We then remove the sugar through simple dissolution with a convective flow, and then we're able to perfuse it with something like a cell medium or blood, or in the case of an intravascular device, the patient's blood. So this is the application for the sugar 3D printing. And now I want to talk about another way you can impart 3D printability in your materials, and that's engineering it in order to be able to have this property that we call a thixotropic property. So that's a property that when a material is being exposed to shear stress, it behaves like a liquid. So shear stress, like the shear stress materials experience as they're being pushed out of the barrel and the nozzle, but a solid behavior when there's no shear stress. So this is what we call a thixotropic material, and how we are able to determine that we have achieved one or that we are studying one is through two moduli. So what I'm talking about is the loss modulus and the storage modulus. The loss modulus is a quantitative description of the liquid-like behavior, and the storage similar, but for a solid. Now, both of these moduli can be measured using oscillatory rheology, which is what we do, or sorry, rheometry, which is what we do a lot in our lab. And essentially what you want to see or what you're hoping for in a bioprintable material is a liquid behavior, so a loss modulus greater than a storage modulus in the nozzle, or when you're exposing it to high shear, or shear equal or greater or less or greater than what or less than what you would experience in the nozzle and barrel, but a storage modulus that's greater than the loss modulus as soon as that shear is removed. Now, we were able to achieve this with alginate. So if you are familiar with our work, we do a lot of work with alginate because it's a great, or it's a really widely studied encapsulation material, especially for immunoprotection. And we were able to achieve thixotropic bioprintable material by modifying the gelling agents. So instead of giving it all the gelling agent in order to fully gel it, which would yield a solid, or not giving any gelling agents at all and yielding a liquid, we end up somewhere in between where we can achieve both a solid and liquid behavior based on the shear rate. So this can be done with a lot of other different hydrogels that cross-link through other means, such as temperature, other cross-linkers that aren't calcium. And we've also shown that we can tune the dynamics of the storage and loss modulus based on how much cross-linking we do. So this is how we characterized our alginate bioink. We 3D printed single multi-layered grids. So as you can see, for the 2%, and we did this with multiple formulations of alginate. So you can see with the 2% alginate, the first layer seems okay. The second one is still okay, but it's starting to collapse. And then once you get to 3, the hydrostatic pressure of the print just overcomes the yield stress of the material, which the yield stress is something you can measure when you measure the storage modulus. So when you increase alginate concentration, you can get a more stable material when you start 3D printing multiple layers. And when you put cells in there, single cells, they seem to survive. So in addition to print quality or the actual geometry of your prints, what is also important in bioprinting is whether your cells survive, so that the cross-linking doesn't affect the viability, and also other things like the extrusion process. And again, if you're familiar with some of our work, we do a lot of work with pseudo-islets or islet-like clusters. And these are cell aggregates, islets are anyway. And in order to mimic this, we also 3D printed cell aggregates similar to the size of islet equivalents, and also achieved some pretty promising viability. So the next case study that we're going to talk about is embedded writing. So this is a different type of 3D printing where instead of the ink having the mechanical properties in order for it to be 3D printable, we have a matrix or a bath that you're 3D printing into, and that bath is providing the support for your 3D print so that your ink doesn't have to have the 3D printability. Now, this is really great because a lot of biomaterials or materials used for tissue engineering are pretty soft or pretty liquid and aren't able to be 3D printed. And this is a great way to streamline non-3D printable materials. It can also improve the complexity of the 3D prints, especially if the bath is not just a support, but also has some function, which is what you'll see in some of our work. And it can also help with creating higher density or more cells, having more cells in your construct. So let's boil down this the embedded 3D printing to the rheology again, similar to what we did with the bioprinting. So before you print, what you want is a matrix that's solid in order to keep the cell suspended. But while you're printing, so as a needle is sweeping through the bath, you want it to make room to displace itself in order to make room for what you are depositing. So this is a liquid-like behavior. And then as soon as the needle is done passing through it, you want the matrix to become a solid in order to keep that structure in place and to keep the cells still suspended. So again, you have a solid, liquid, solid, quick change in properties. So unfortunately, we're not able to disclose what our matrix is or how we achieved it, but essentially we're able to create different formulations of our material where we can achieve different levels of printability. And we're also able to show that based on printing parameters, you can get different diameter filaments. And again, when characterizing bioinks, it's really important to see or to determine whether your cells are compatible within it. So this is some viability data of our formulation B, showing that we have good viability. So hopefully you have a good idea of some of the applications and the principles of creating a 3D printable platform. And I'll pass it off to Professor Hoesli to wrap us up.
So in summary, we showed you some of the applications of 3D printing and encapsulation in medicine. So some of those were covered today, but it's a very broad field, as I mentioned earlier. I encourage you to read some of the review papers and some of the papers that were in the references in the video to learn more about this. We spoke about some approaches, categories of ways to generate living tissues, layer by layer, in situ casting. We also talked about the 3D printing, the actual equipment type categories of how you can deposit living cells on a 3D printed construct. So by extrusion, laser or droplet technologies. Brenden covered some concepts in 3D printability, relating printing parameters to the filament dimensions that you're getting, and also how the rheological properties of the material can be used to predict and to optimize your prints. And then finally, we showed you some applications by which you can generate vascularized tissues, either by the casting of sacrificial materials, or the second case where Brenden presented some ideas on embedded writing to be able to print and to use materials that are harder to print by more conventional means. So using in situ type applications. And finally, I want to thank our collaborators on many of these projects. So some of them were already mentioned in the talk, but I also want to thank Greg Corbett and Tim Kiefer, with whom we've been interacting to test out the tissues that we're printing. So with that, thank you. And we look forward to any questions. And if you don't have time to ask them during the webinar, you can always email us. Thank you. Thank you for joining. So if you're joining just now, we just finished a portion of this webinar that was on the health aspects of 3D printing. And now, as the second part of the webinar, we will be talking more about agricultural applications of 3D printing. And this portion of the webinar will be presented by Craig Bundt at the University of Otago, New Zealand. So Dr. Bunt is the inaugural professor of agricultural agricultural innovation in New Zealand. He has a background in pharmaceuticals, and he has extensive experience both in academia and in private industry. And he would also obviously be very happy to entertain any questions. If you don't have time to post them in the webinar, you can always contact him at the email that's provided on this slide.
Hello and welcome to my talk. So I'm Dr. Craig Bundt, Professor Craig Bundt, newly appointed at the Otago University in New Zealand. Prior to this role, I was an associate professor in animal science at Lincoln University, still in New Zealand, and have been a government scientist in agriculture, lectured in pharmaceutics, and a research officer for a plastics injection molding company. And where I'm getting to with this is pinpointing where my interest in something that eventually became 3D printing started. I did my PhD in pharmaceutics on microencapsulation of bacteria. And after that, went and worked for a company on plastics molding of drug delivery inserts for an agricultural product. And it's out of these two areas of interest that my focus now in 3D printing and encapsulation bears some responsibility from this time many years ago. Now, why 3D print encapsulate for veterinary and wildlife and agricultural applications? Well, additive manufacturing, this is the layer by layer, piece by piece, precise assembly of constructs, is what additive manufacturing is. It has a number of advantages. And currently there's 7 types or families of technologies, but this is so rapidly evolving. I wouldn't want to answer a question in a quiz, how many types of additive manufacturing are there? New ones arrive weekly, not weekly, monthly. The advantages of additive manufacturing includes you can make things at small scale that traditional manufacturing techniques would not be able to address. The quite complex tool construction you need for injection molding is a significant cost up front, in the tens of thousands of dollars to hundreds of thousands of dollars, depending on the complexity of what you're making. Also, conventional manufacturing in terms of just mixing in a pharmaceutical scenario, we know the challenges of going from small vessels to large vessels and scale up as an exercise in its own right is challenging. So if you're never needing to go to large scale, or you need to assess something on a small scale first, then expensive equipment is going to be a significant cost. But with the additive manufacturing, the costs, particularly for changing things, trying out new ideas, aren't a significant barrier. Additive manufacturing also gives you precision on where you place things, quite good precision in terms of the dimensions of complex constructs that something such as conventional injection molding might not be able to do. Don't get me wrong, conventional injection molding can be incredibly precise, but there are limits in terms of how you can design a mold to be able to accomplish that. And additive manufacturing gives you the advantage often of a continuous manufacturing process rather than a batch to batch variation that you get in conventional manufacturing. The loading and unloading and reloading between batches and conventional mixing. It's also incredibly adaptable. And you can make changes in the design phase repeatedly, quickly. It's a very design phase heavy process, as opposed to conventional manufacturing, which is very heavy in the equipment setup phase and giving you very little opportunity for redesigning and tweaking products. And so additive manufacturing really enables the ability to rapidly redesign and change without having barriers where we can't change the mold anymore, or we can't even mold that. So this adaptability makes it very attractive. So One type of, I regard this as additive manufacturing, 3D printing technology that wouldn't typically be called 3D printing, and that's membranes emulsification techniques for microencapsulation. So forcing a material through a size dependent restricted membrane to create very precise microparticles, microencapsulation, by different variations, giving very tight particle size distributions, very uniform microcapsules, as opposed to anybody who's done typical beaker, vat, vessel, solvent evaporation, emulsification. You can get some very ugly variable constructs more often than not. So this ability to essentially assemble each microparticle independently of the bulk material, or to process a coarse emulsion in this case into small, discrete, defined particles, and not in one step, as a continuous process. It gives you precision. And I regard this as an additive technique because we're only assembling some of these microcapsules at each time point. It's not a technique that's producing everything at once and introducing variability. And in agriculture, there, or particularly in veterinary pharmaceuticals, there's an application, one of many, of course, where precision of what you're, say, delivering in terms of a therapeutic is absolutely crucial. And one is if you're trying to reproduce a natural process where progesterone drops off in an estrous cycle of cattle and other mammals. And currently, the only way that's used commercially to do that is through the use of a large silicone insert that contains progesterone. And the way you mimic progesterone's drop off is by removing this from the animal. And it contains a large amount of unused drug. It's expensive to manufacture. It has been shown, and some time ago, that if you have very precise particle size, you can get very precise delivery and drop-off of drug, particularly progesterone. And from a single injection, then deliver everything you need. But as I mentioned, this requires precise, uniform particle size distribution. And if you're using conventional, typical microencapsulation techniques, you might not always be able to achieve this. And this is some work from quite some time ago. This is the sort of application in veterinary agriculture and veterinary pharmaceuticals that would benefit from a precise step-by-step assembly of microcapsules to control that particle size and other properties that will then dictate your release. Something that I feel definitely meets the definition of additive manufacturing is electrospinning, electrospraying. It's a layer-by-layer technique with a solution or a melt of polymer containing drug fired from a needle or a rotating disc to a target earthed collector. This enables you to deposit fibers or particles, droplets that are on fibers. You can get quite creative with what you're building up, and you can build up layers of material. These fibers can contain drug or other bioactives. Certainly these particles here would be quite significant reservoirs of any bioactive. And as I mentioned, you can build up layers of different material. You could start with a water-soluble layer, spin a fiber particle layer, or just a fiber layer of insoluble material, and build up layers for whatever application you might have. Excuse me. I've looked at this for literally spraying onto, in this case, cabbage seedlings by earthing, pun intended, the cabbage seedling and spraying a charged polymer stream towards the plant. You can coat the plant in electrospun nanofibers. They don't appear to interfere with the growth of the plant. We used a water-insoluble polymer here. I won't disclose what we used, but they will crack and break as the plant grows, so they don't interfere with plant growth. This was a solvent-based electrospray. The solvent had evaporated by the time the fibers had deposited, so we didn't damage the seedling. And in this case, we had loaded into the nanofiber neem oil. Neem oil is our anti-feeding organic insect repellent, essentially. And so by spraying cabbage seedlings, in this case here with this neem oil containing a nanofiber against one that was not coated, you can see this one is significantly damaged by caterpillars that would, when released near the seedling, migrate to the uncoated seedling and damage it, and were repelled from the coated seedling. This is also water resistant, shower resistant, rain resistant coating, and it physically traps the neem oil on the plant. So there's a number of advantages here. We're not washing bioactive off into the soil, we're keeping it where we need it. There's no point applying material where you don't need it. So you've got precision application. This is one of the advantages of electrospun nanofibers. They will land where the earth is. So the plant being the closest earth to the fiber production needle gives you fibers landing on the plant. So quite a nice little agricultural application there. This can be done at scale. It can be done out in the paddock with appropriate constructive equipment. It can be done in a glass house, and in this case in the laboratory. But when we talk about 3D printing, most of us would think of the layer by layer FDM or FFF. Both these terms are describing the same process. Fused deposition modeling is typically thought of as a more industrial scale, more industrial precise process. And fused filament fabrication, FFF, is usually thought of as the more home hobby level of finish, so to speak. But both these processes are essentially the same. A hot melt is deposited layer by layer, and the construct is built from the bottom up, with other support things built along the way. So you can build things that you would then put something else in, or the filament that you are depositing here layer by layer can contain bioactives. And so some research I've been involved with has looked at loading the polymer and then printing it, in this case here, into darts for firing out of an air rifle for delivering a bioactive to wildlife. Now you might ask, why would you want to do that? Well, some areas of interest in New Zealand centres around protecting our wildlife, and this is not common to New Zealand. We have unique wildlife we'd like to protect, and that's usually protecting wildlife from predators, and that's the typical situation here in New Zealand. We have some of the world's largest insects. Don't worry, they don't bite. We, of course, have the kiwi, which I'm sure you're all aware of. In New Zealand, we also have the world's largest flightless parrot, up to 4 kilos. And interestingly, the one land mammal we have is this tiny little bat. You can see a human thumb here, I think. A very small bat. Nationally, we've just voted it the bird of the year. So a little bit of a interesting winner there, but it's certainly drawn a lot of attention to this very rare endangered species. So wildlife protection, wildlife solutions, wildlife control is getting increasing attention. One of the challenges here, though, is what you might be delivering to wildlife, be it therapeutics to endangered species, or toxins to other predators and pests and such. You face the challenges of many, many different configurations, many different uses, many different targets. Sorry again for another pun. But also short production runs or limited amounts of material needed, or the need to be able to rapidly adapt and change as how these sorts of things could be used or are used, changes within more science to support their use. So in New Zealand, we have a native mountain parrot, the only one in the world, and it's evolved away from mammals. So when this introduced pest gets in to its nest, it won't defend itself. This pest can come in and take chicks, takes eggs, and takes them away. And this parrot here, which is the size of a very large seagull, with this can opener-like beak, puts up no defense. It's trying to rip a car tire here, and it can probably do it, yet it has no mechanism to defend itself from these. So we might be interested in delivering, say, birth control agents to this possum by a bullet approach. But the costs of setting up a production facility using current technologies for that would be cost prohibitive. And this is something a program in the US struck with trying to control a disease in endangered bison in Yellowstone. They typically can catch a disease that can spread to cattle and elk. And so to treat these animals and protect them from this disease, a vaccine is needed. But how you vaccinate wild animals in a national park is challenging. You can't use conventional vaccine darts that would leave bits of plastic and needles and metal lying around. You might miss. You can't cage them all. These are incredibly large animals. So a biodegradable bullet containing a vaccine would be ideal. But using conventional manufacturing techniques, it's cost prohibitive. And so this was looked at some years ago. It was understood that a biodegradable bullet to deliver vaccines would be required, but the technology at the time wasn't essentially cheap enough. And so the program had to be abandoned. So the program was going to use this thermoplastic injection molded hydroxypropyl methylcellulose biobullet, so biodegradable thermoplastic, each one individually formed in a mold or a mold with many cavities. Every time you might want to change this here to larger or smaller, you would have to produce a new mold or modify an existing one. And also only a company owning a mold could manufacture these. Another company, if they were going to manufacture them, would have to invest in the mold. And these tend to be tens of thousands of dollars as just a part of a machine that's going to cost hundreds of thousands of dollars. These bullets here have a hollow cavity, and in this case here, they're filled with a hydrogel containing a vaccine, and then fired from a pneumatic rifle, so an air rifle. So there's a lot of bottlenecks there for the development and the uptake of this sort of technology. And certainly as this program would, if it had been carried out, if it would have met with success, the need for these each year would drop off. So anything in wildlife control, it's anticipated year on year, you wouldn't need as many. So who's going to invest up front if you end up destroying your own market? We have similar challenges in agriculture as well. For veterinary pharmaceuticals, there might not be enough need or enough numbers to justify the expense of the treatment. And so that can be a barrier to developing new technologies. Now with computer-aided design and 3D printing, you can get very creative up front with designing new bullets, with designing new delivery abilities at very, very little cost. These biobullet designs and the 3D printing is a process that is computer-based, so it's just time. You could spend quite considerable time coming up with as many crazy ideas as you can imagine. A solid bullet, where the drug would be incorporated into the polymer prior to forming, or various constructs with cavities that you can put an insert in that contains your bioactive. And so you've got adaptability, flexibility. In this design process, you can go through that before you go anywhere near actually making what it is you're hoping to make. And so if you were to be successful and no longer need to make these for one application, you can redesign something else and use the same printing equipment to make something entirely different for a different application. Whereas the mold to make these is only ever going to make those. And every time you want to change something, you've got to make a new mold. Here, every time you want to change something, you just design something on the computer. And so these bullets, these darts containing, in this case, progesterone loaded in the polymer prior to molding, releases as you would expect for any monolithic device containing a drug in a polymer. And so for very little equipment cost, you can design something and print it. So the blending of the drug and the polymer is going to be conventional plastics blending equipment, and the printing is going to be anything from a home hobby printer through to an industrial precise 3D printer. You can print one insert as easily as you can print 100 at a time. And so scale can be influenced, changed very easily. There are things about 3D printing that can give you functionality that conventional molding won't as well. This layer by layer printing process for FDM or FFF printing comes through in the final product. So the orientation that you have of your device when printing will influence its structure. So if this dart is printed layer by layer up through the process, then you see these layers appear in the final product. If this dart was laying horizontal 90 degrees, then you'll have, when you turn it upright again, you'll see layers going across, and you can have layers going this way. Layer orientation then influences how strong the thing is that you have printed. Where your layers are your weak points, to be honest. So this dart, same design, but the orientation, if you print it vertical, so standing up like a rocket, the force to break it is about 200 newtons. The same dart, if it's just printed 90 degrees on its side, the force to break is about 350. And that can change the ballistics of what you've got. It can also change the characteristics of when it hits its target, whether that stays intact or breaks off. And breaking off can be a good thing. Maybe you only want the front of the dart to break off and deposit a payload, and the back end breaks off and drops away. And maybe only the front part contains your bioactive, because you can print different regions of different material. And like I say, just depending on the way around that you print it, you can influence the performance characteristics and get some added functionality. So never underestimate the importance of orientation when you're 3D printing. You can get it very wrong and create a fragile thing, but you might get that very right if you want a fragile thing. Vice versa, it will influence strength as well. And again, depending on the drug loading in the polymer, we can influence release rate. So not only can we adapt release rate based on drug loading, we can influence its ability to break or not, depending on the orientation. And we can anticipate then that something that breaks up will release its payload quicker. We haven't done that work though. That's all I'll really say about 3D printing on this small scale for delivering bioactives. I'll just finish up now with some comments on why I think every laboratory should have a 3D printer for many, many applications. Never underestimate the ability to rapid prototype. If your lab doesn't have a 3D printer, just a home hobby one for a few $100s, you're missing out on a lot. Find somebody in your lab who's young and can Minecraft, and they can have this up and running for you in a day. You can then maybe find something in the literature that you think that's worth investigating, and it's just an idea somebody published 20 years ago. You can CAD and print it up, and 2 hours later have a working physical model of what this thing here was proposed in a patent many, many years ago. You can work with a piece of test tube you've just cut up to create a speculum that you're going to need, and in this case, for some work with young sheep, and go through a second prototype, a third prototype, and a fourth prototype by the afternoon, and have something that's got significant improvements on the original test tube based model in terms of reduction of material by having hollows in it, voids in it, and functionality for what you need. And if we'd gone through a conventional manufacturing process to get to this point, rather than 4 hours, 5 hours, it would have been 4 or 5 weeks and tens of thousands of dollars. This is time and $2 worth of polymer. 3D printing also enables you to modify very easily existing technologies. This is an old piece of equipment consisting of a blanket, a unit strapped onto the back of the animal that could measure urine frequency and volume. And this is information we need to know in terms of mitigating climate change, how much nitrates are put out by a cow and when and where. You can't go following the cow with a bucket around the field. How do you do that? You measure the output. This whole setup here weighs kilos. By looking at where we could cut away weight, where we could produce things on a 3D printer that we couldn't produce any other way, catting these up and printing this flange, we could attach this to the back of the animal with a simple strap and some sports tape. This will only stay on the animal for 2 days, measure urine frequency and volume as it goes through a sensor, and then fall off the animal. And because each one of these only takes a few dollars to manufacture in terms of materials, if it's damaged by cows stepping on them, that's not an issue. Added advantages, we can make that out of a biodegradable material, in this case we have, and so if it's lost on the paddock, it'll just rot away in the compost. So we went from a many kilos device, and this one here a few hundred grams, in a very short, quick process. If you're interested in things like this, you can actually find this design on the Thingiverse and just search my name. And just to wrap up, 3D printing, it's great for clinical models as well. Honestly, this is, I have to let you know, that's my jaw from a CT scan, and that's a print. And this was done prior to a dental implant. And so while the dentist and the dental technician was only working off this, I felt I wanted to print my entire set of jaws and get a really good idea of where the implant was going. This then led us to some work where we took the CT scans of sheep that had a brain shrinking disease, genetic deformity, and taking those CT scans through to clinical models. In this case here, a sheep that's had it's a normal sheep, normal brain size, CT scanned, back out into the paddock. This sheep here has the brain shrinking disease, and we can see it's a much smaller brain cavity, and we can use that in clinical teaching and education. And again, my set of jaws and sheep's brains, if you ever want to print one, can be found on Thingiverse as well. So that's all I have to say. This slide just to remind me to stop talking. Thank you for your time. I hope I haven't bored you. And if we've got any questions, hopefully some questions, we'll answer those to the best of our ability. Thank you.
So thanks for those presentations. And so we're happy to take some questions. So I believe some of them have already been answered in the Q&A, but this is a good time now to post any other questions you might have. And Brenden and Craig, you can open your mic and camera. So Craig, I had actually a question for you. So with those bullets, those vaccine release bullets, you compared the mechanical properties with the horizontal versus vertical prints, but how does that compare to a casted bullet?
If it had been cast, I would have imagined that sort of unit would have taken probably close to a kilo to break for that small size unit.
And when you measure
so probably about 3D printed or maybe a third the strength of a molded unit.
When you measure that, in which direction do you measure the mechanical properties? Like, is it by compression, or how do you test?
Yeah, so for the bullets, it was across the cradle and compression. with the blunt blade.
Okay. And I was wondering also if it changes the actual ballistics, the fact that it's 3D printed, or just upon impact. I know it's not something easy to measure,
Yeah,
but
these are, yeah, I did some. It's a good day in the lab when you get to cast ballistics gel. They're not very accurate. The downside is all these materials are incredibly light, and so we started investigating adding some dense iron, things like that, zinc oxide, to improve the ballistics. So the major issue at the moment isn't that they're 3D printed, it's just they're too light.
I don't see
For an air
too
gun.
many.
If it went to a firearm, we might get around that, but there's a lot more paperwork at the university to do that.
I don't see too many questions in the Q&A. There's a couple of questions that were already answered. Okay.
I have one for my colleagues here presenting in terms of tissue culture, tissue engineering, regenerative culture. Do you talk of clean meat in the lab much? Being in agriculture, it's a conversation we have a lot.
Oh, the meat. Yes. Okay. So we've been contacted by companies that are interested in producing muscles from stem cells for that application. But when you work with the kind of cells we work with, the costs are very high. You know, like one burger would cost hundreds of thousands of dollars. People are trying to bring this down by using other sources of growth factors. If you want to make it cheap, sometimes you would use a bovine serum, but then you're back into using animal-derived products. So I think personally the economics, if you want to do this from stem cells, it's going to be very tricky. And if you do a sustainability analysis, I'm not sure this would turn out to be beneficial for stem cell derived products. But I do like some of the other ideas out there, like genetically engineered yeast, or basically just sources of proteins through cell culture, but not necessarily from stem cells, although apparently some people have been able to solve some of those cost issues by finding, like I said, other sources of these growth factors that are less expensive than the ones we typically use. But it's definitely a field that's emerging. I would just encourage anyone that's jumping into that field to do a complete life cycle and sustainability analysis.
Yeah, and if I could jump in as well, I think aside from the stem cell stuff, the 3D printing, I think is an avenue that some researchers are looking into, not just for the cells or the cell culture, but more for like the texture of the food, like recreating marbling, for example, in meat. So a lot of mechanical properties associated with eating and the meat that people have strong feelings towards, they're trying to recreate using 3D printing.
I think those scaffolds, there's a lot of research there, opportunities for people. I totally agree with the non-stem cell approach for alternate proteins, particularly those that then can be processed to meat-like. What amazes me is every company that says it's sold for stem cells got around the use of bovine serum and such. They never then mention antibiotics and how much they are using it. And there's, we need to use less antibiotics in agriculture. Clean meat isn't necessarily going to do that. And I would say you would never try to grow stem cells without antibiotics in your lab. Am I correct?
No, you do actually, like it's at least with the human stem cells, we typically grow them without antibiotics. But it's true that if you're manufacturing at scale, the risks of a batch getting contaminated might be high. But actually, there are a lot of labs that are not using antibiotics. So
you're using aseptic techniques.
Yes, exactly. It's completely aseptic. Yeah.
Yeah. So then how's that done at scale? Yeah.
Maybe we could still go over some of the questions that were already answered in the Q&A. Maybe not all of them, but I think some of them are quite interesting. And when the webinar is posted, this might be useful to have the actual oral, like the spoken answer. Anand Patel is asking, when you add calcium salts to prepare your thixotropic alginates, how does that affect osmolarity and viability, Brenden?
Yeah, so that's a really great point. I think when you're modifying hydrogels or any sort of aqueous solution, the osmolarity definitely has a role. And we've seen not properly handled solutions cause death to cells based on osmolarity. So we have not looked at it in the research that we presented. However, this is something that we are very interested in looking at. We do modify we have theoretical values of what the osmolarity should be based on the chemical structure and the dissociation of some of the salts we use. So we aim for around an osmolarity of around 300, 310 milliosmoles for all of our solutions. And when we combine them, we assume that the osmolarity stays in that range. But definitely something we can look at. And yes, we've only been looking at the performance of our inks using viability. And we've actually not shown here, but we've cultured cells that have been 3D printed in some of our inks for multiple days and see some pretty good viability and functional data as well.
Yeah, and then perhaps Craig could take this one. It's already been answered, but realistically, how small can you actually go with 3D printing? How thin can you make your filaments?
With fused deposition, it can be 200 microns, but with scintigraphic techniques, a few molecules. Really it is how much money you want to fork out on equipment. But my rule of thumb is the smaller you want to go, the less you have in terms of options for polymers. You might be using some sort of polymerization technique, and then you have issues with residues. If you're already using ready formed polymers in a melt deposition type process, high nano size in terms of thickness, you can certainly do that with spraying. But if you wanted to get individual microcapsules 3D printed and build up a shell, that you could do it. It would be fun, but I don't think you would do it very often.
And on the bio ink side, some of the technologies can allow you to go very small, like the laser or inkjet technologies, for example. Rene Pedrosa is asking, when printing cells versus pseudo islets, is there a difference in their distribution along the filament?
So yeah, another great question. I think when I answered it, I more talked about in the radial direction, but along the filament, we haven't seen any sort of differences between printing cell aggregates and single cells in the work that we have done. But there could be ways that you could control the position of your cells based on your extrusion. So if you are looking along the z-axis of a cylindrical coordinate, then perhaps the ink loaded into your cartridge could be more modular, more not uniform. So in order to get something like that, we don't really do a lot of that work, but this would just be an off the top suggestion. If you are trying to position items such as cells radially, you can use a coaxial nozzle, which has been my answer to a couple questions in the Q&A. But essentially, if your nozzle has a shell and a core, and we've even seen like triaxial nozzles and above, so multiple shells in a core, you can position things, materials, cells in the radial direction. So creating a core, a shell, an inner shell, outer shell, and things like that. But you will have to worry about your materials mixing as you are extruding. So something that you can look at in that case is adding gelling agents in your different layers that would gel the counterpart layers.
One interesting idea that I think could be explored, I don't know if anybody's done it, but basically use a kind of imaging technique, like so they can track when particles are coming out of the nozzle to adjust your printing parameters. It would be really interesting to have more feedback control algorithms on 3D printing, and that could address maybe some issues when you're printing larger particles like pseudo-islets. Just an idea, throwing that out there for anyone that's working in the field. And the question for Dr. Bundt, I know it's been answered already, but what about the prices of electrospinning, especially for agricultural applications?
You take away the word scientific off a piece of equipment and it drops in price by an order of magnitude. So a bench top electrospinner is going to cost us $20,000 for scientific equipment. You can build that bespoke for $2,000. You know, there's a comparison of scale there. So I have seen units constructed for on the back of a tractor for spraying directly onto pasture. It's really some pumps, your material. With electrospraying, remember you're applying not very much material at all. In some coating scenarios, you might be only adding a fraction of a percent of material to your bulk. And the high voltage generator, you can source those for $1,000. Really, the limit then is just how far an extension lead you can run out into the paddock. So I don't think there's a cost issue. And also we're now seeing nanofibers built, manufactured at large scale for air filtration for buildings and disposable face masks. And so if we can compare those, the cost of that is very comparable to what agriculture could tolerate.
All right, so I think I will end the Q&A period here, but do not hesitate to email us if you have any further questions. And hopefully we covered some of the major questions that were in the chat. So with that, I want to conclude this event by inviting you to look into future BRG events that are upcoming in 2022. So there is an industrial convention in Rotterdam in May, and registration is already open. I invite you to attend. These conferences are fantastic if you want to network with industrial partners, and you can have one-on-one meetings with companies that you're interested in collaborating with. There is the 27th International Conference on Bioencapsulation. Stay tuned. The registration is not open yet, but it's coming up shortly. And then upcoming webinars, including granulometry, microencapsulation, characterization, Pickering emulsions, and other techniques are coming up. So go on the bioencapsulation.net website to register for the upcoming webinars. And you can also watch the past webinars that are recorded online. And this current webinar will be posted shortly. Thank you for joining, and see you soon.